through hydrogen bonds, while cytosine pairs with guanine through 3 hydrogen bonds. So apart from this DNA, we have one more thing, that is triplex DNA. So in the year 1975, G. Felsenfeld and his team observed this unusual and fascinating DNA structure known as triplex DNA. So it is also called as triple helix. So here this structure is formed when the third DNA strand inserts itself into the major groove. So here we can see the light green color shade, it is a double helix of DNA. So here in the major groove, we can see one more strand. So this is a strand 3, so which inserts itself into the major groove of the DNA strand. So for this type of arrangement, for this type of arrangement to occur, so here the double helix, what we see here in the green color, so must be somewhat asymmetric with one strand. So out of these two strands, one strand must be rich in pyrimidine bases. So, and the complementary strand, that is the third strand, should be rich in purine bases. So here this asymmetry is critical because it allows the third strand to form specific hydrogen bonds, that is known as Hoogsteen hydrogen bonds. So with purine rich strand. So this is the purine rich strand. So this is the triplex form of DNA. And we have one more type of DNA, that is G quadruplex. So apart from well-known double helix and triple helix DNA structures, DNA also adopt one more fascinating non-canonical conformations, that is we call it as G quadruplex or G4. So shortly we can call it as a G4. So here this structure arises in guanine rich sequences. So here we can see this is a guanine rich sequence. So where guanine bases have the unique ability to pair with each other. So these guanines have the unique ability to pair with each other, so through Hoogsteen hydrogen bond. So here we can see, so 4 sets of guanines have been bonded together, linked together through Hoogsteen hydrogen bonds. So these are stabilized by a monovalent cation like potassium or sodium. So this set is called as a G tetrad. So when these G tetrads are stacked on one another, it forms a G quadruplex. So this is the G quadruplex. So here these structures are not just structural curiosities, but they are functionally very significant, as they are often found in biologically important regions such as telomeres, genome stability, and even in potential therapeutic targets in cancers and other diseases. So coming to orientation of these strands. So here the orientation of the G-force, there are mainly 3 types of G-force as how these strands run. So there are mainly 3 types. One is parallel G quadruplex, second one is anti-parallel G quadruplex, and third one is hybrid or mixed G quadruplex. So coming to parallel G quadruplex, you can see here in the image, that is in parallel G quadruplex, all the strands run from 5' to 3' direction. So this is it has a very uniform structure. And coming to the anti-parallel, so here in anti-parallel G quadruplex, some of the strands run in 5' through 3', whereas some strands run in 3' to 5' direction. So that's why it is called as anti-parallel G quadruplex. So coming to the next one, that is last, hybrid or mixed G quadruplex. So this hybrid or mixed G quadruplex is nothing but it is a combination of both parallel and anti-parallel strand orientations. So G quadruplexes, G-force are common, are not randomly distributed in the genome. So instead, they tend to localize in biologically important regions where they play crucial roles or regular roles. So one of the most important prominent sites is promoter region located just before the start of the gene. So here one of the most these regions are often guanine rich, and they can easily unwound themselves, which are favorable for the G quadruplex formation. So interestingly, it has been noted that around 40% of the human genes harbor potential G quadruplex structures in their promoters, particularly in oncogenes or cancer promoting genes. So, and one more key location is in the telomeres, so which are guanine rich repetitive sequence at the end of the chromosomes. So here G4 structures act as protective caps, shielding DNA from degradation and damage during cell division. So here intramolecular G4s have also been identified in several important genes. Some of the examples are that is CMYC, CMYC, c-KIT, BCL2 in HIV genome. So this is often in human telomeric sequences. These G4 structures they can block telomerase enzyme. So G4 stabilization and detection. So here as we know, the G4 structures are non-canonical secondary structures of DNA or RNA. So here bioinformatic analysis of the human genome have revealed the presence of more than 7 lakh potential G4 forming sequences, highlighting their abundance and potential significance. So here, because of these structures are not just structural oddities, they are believed to regulate some of the key biological processes such as transcription, translation, and the maintenance of genomic stability. So because of their importance, so there has been intense interest over the past decade in developing fluorescent probes or small molecule ligands that can specifically and selectively recognize these G4s, and they can stabilize them. So most here these fluorescent probes are the small molecule ligands. So these are polycyclic planar compounds that interact with the G quadruplex sequences primarily through pi staking on the G quadrats and through electrostatic interactions with the phosphate backbone. So here, for these ligands to be effective, they must display high selectivity for G quadruplex sequences over canonical DNA forms like single-stranded DNA, double-stranded DNA, and all. So to study and confirm these interactions, so we use a range of powerful techniques are used, like UV visible spectroscopy, circular dichroism, fluorescence assays, and NMR, that is nuclear magnetic resonance. So coming to applications of G4, that is in the field of biological significance. Here, to begin with, these G4s are highly enriched in the promoter regions. As said, that is, they are highly enriched in the promoter regions of several oncogenes, such as some of the examples are CMYC, KRAS, and BCL2 G quadruplexes. So where these act like a molecular switches that can either suppress or enhance the gene expression, thereby directly influencing the gene regulation. So moving to telomers, which are guanine-rich protective caps at the end of the chromosomes. So here these help maintain chromosome stability by shielding the DNA ends from degradation and inappropriate fusion events. So importantly, they also serve as natural inhibitors of telomerase, that is an enzyme so enzyme that is abnormally activated in most cancers to support uncontrolled cell division. So making G4s a potential anti-cancer target. So beyond transcription and telomere maintenance, G4 also play critical role in that is DNA replication and repair process. So they act as regulatory barriers that modulate replication, origin firing, control the movement of replication folks. And in the field of diagnostics, so here these G quadruplexes play a very important role as biomarkers, because these G4 motifs are enriched in regulatory regions of oncogenes and telomeres. So their presence and stability closely link with the diseases such as cancer and neurological disorders. So this makes them promising biomarkers where detection of G4 formation or stabilization in specific genes can serve as indicator for a disease and onset or progression. Coming to therapeutic potential, so the therapeutic potential of G4s is also exciting. More exciting, where in the field of biomedical research, so as these structures offer novel strategies for targeting diseases at the genetic and molecular level. So in the context of cancer treatment, so here G4s play a pivotal role since they are highly enriched in the promoter regions, so as well as in telomeric DNA. So by stabilizing these G4 structures with small molecules or fluorescent probes, so we can suppress the oncogene suppression and inhibit the telomerase activity. So effectively limiting the uncontrolled growth of cancer cells. So beyond oncology here, G quadruplexes are also emerging powerful targets in the field of antiviral strategies. So many viral genomes, as we see, that is HIV, SARS-CoV-2, hepatitis virus, they all contain G4 forming sequences and that regulate viral replication and gene expression. So here, by stabilizing or disrupting these viral G quadruplexes, it is possible to hinder viral proliferation and reduce infection. So in the field of drug development, so these G quadruplexes have emerged as highly attractive targets because of their selective presence in biologically critical regions such as oncogene promoters, telomeres, and viral genomes. So their unique structural features, staked guanine tetrads stabilized by cations, makes them distinct from the canonical double helix, allowing the design of small molecules that can selectively bind to a particular G quadruplex sequence. So which makes the target identification very easy. Uh, so G quadruplexes also play a very important role in understanding the genetic disorders and genome stability. So because of their unique ability to form stable secondary structures in guanine-rich regions. So here, in this case, G4s or G quadruplexes can act both as protectors and disruptors of the genome. So coming to methods of detection, so one of the most widely used approaches is based on the fluorescent probes. That is where these we use small molecules or ligands. Where these molecule fluorescent probes bind to the specific G4 selectively, and there will be a strong fluorescence signal allowing real-time visualization of G-force with respect to fluorescent probes. And another two methods are electrochemical methods and uh, NMR and X-ray crystallography. So coming to quinolinium probes for G4 detection. So here in our work, we have used quinolinium based probes for the detection of G quadruplexes. So here among the many fluorescent tools available for studying G quadruplex sequences, so quinolinium-based probes have attracted attention due to their salient features like strong binding affinity, selectivity, and excellent photophysical properties. So here these probes are typically typically cationic aromatic molecules with a quinolinium core that intercalates or stacks with the G tetrads or in the G quadruplex. So here upon the binding of this cationic species to the G4, so they exhibit a remarkable light up effect, that is the fluorescence will be enhanced significantly, providing sensitive readout so for the presence of these G quadruplex structures. So importantly, many quinolinium derivatives can differentiate G4s from duplex DNA and single stranded DNA, reducing background signals and enhancing selectivity index in complex biological environments. So here we can see the molecules. That is, we have used two molecules for the detection of G quadruplex. That is, one is QNMF. So here QNMF was synthesized using 1,2-dimethyl quinolinium iodide with the morpholine derived benzaldehyde derivative. So we got a molecule that is QNMF, and the second one was that is QNA. So here the N-methylated benzaldehyde was treated with 1,2-dimethyl quinolinium ion to give the molecule that is QNA. So these are the oligonucleotide strands sequences that are used in the current study. So here we can see that is these are the oligonucleotide names, that is CMYC. These are the sequences, guanine rich sequences, that is from 5' end to 3' end. So this is intramolecular parallel in nature. So even CMYC is parallel, CKIT1, CKIT2 are parallel in nature. So TBA 22 Ag are parallel in nature. So here it is interesting to note that this 22 Ag in the presence of sodium, it forms anti-parallel nature, but in the presence of potassium, it forms a hybrid nature. So even HRAS bomb 17 are anti-parallel in nature. So apart from this, we have used ssDNA and dsDNA, that is single stranded DNA and double stranded DNA for our current study. So the photophysical studies of the compounds were evaluated using different solvents of varied polarity. So here we can see the absorbance and emission of the compound probe QNA. So here the quinolinium based molecule, that is QNA, exhibits pronounced solvent chromism with the absorption maxima ranging from 467 to that is in aqueous media to 528 nanometer in DCM, and emission maxima ranging from 555 to 617 nanometer. So here there are larger soap ships are observed. So in polar and hydrogen bonding solvents such as DMSO, glycerol, and water. So indicating strong stabilization of the excited state through dipolar relaxation. So the significant rest. So here we can see there is a significant red shift in viscous glycerol. So this is glycerol. So which is due to viscosity dependent emission. Similarly, the for photophysical studies of uh, QNMF, another molecule QNMF were also evaluated. So the absorption spectra of QNMF exhibited a hypsochromic shift, whereas the emission spectra of QNMF showed the bathochromic shift, that is a red shift from 550 nanometer in DMSO to 618 nanometer in DCM. So in polar solvents, these solutions were weakly emissive, so which might be due to the formation of J aggregates. So further, we have calculated the quantum yield of the compound QNMF in two solvents, that is from in methanol and glycerol. So here it can be noted that the quantum yield of QNMF in glycerol is around 0.20, which is around 740 times lower than that of methanol, which is the value is 0.0027. And further, the brightness of the uh, QNMF in methanol was very low with a value of 40.5, whereas in glycerol it was very high with a value of 2800. So this might be attributed to the fact that here the viscosity, so methanol is least viscous and glycerol is highly viscous solvent. So this this is due to that is increase in the viscosity restricts the rotation of the carbon-carbon bond around the aromatic units. So this leads to the increase of the electronic movement. So in support of this, we have uh, carried out a viscosity dependent fluorescence studies to understand the restricted intermolecular rotation of the compound. Sorry. So here, this approach is often used to gain insights into the local environment of the fluorophore or the molecule in the solution. So here, glycerol is a highly viscous solvent. So for that purpose, and for this purpose, it is used in such studies, and it can, because this highly viscous solvent, that is glycerol, can significantly slow down the molecular rotation, making it ideal for investigating the effect of viscosity on fluorescence. So here we have examined the fluorescence properties of that is QNA in various viscosity mixtures by altering the ratio of methanol to glycerol. So here we can see the image, that is this is 0% glycerol and 100% methanol. So the percentage of glycerol was increased, the what the emission was enhanced under UV light. So here from the fluorescence spectra, we can see that so this is 0% glycerol, and as the percentage of glycerol was increased, so there was significant enhance in the fluorescence intensity, as well as there was a bathochromic shift, that is red shift at around 580 to 570 to around 618 nanometers. So in the fluorescence. So further, for the viscosity sensitivity of QNA was calculated using the Foster-Hoffman equation. So here we can see which exhibits a linear response. So here the viscosity sensitivity of the molecular rotor QNA was determined to be 0.41 compared to the other traditional FMRs. So hence, due to this strong viscosity sensitive properties, so this QNMF can be utilized as fluorescent probe to detect G4 structures, as it effectively senses changes in microenvironment. So further, we have screened the synthesized compounds, that is QNA and QNMF, for the detection of various G quadruplexes. So here we can see the G quadruplexes over here: 22 AGG, BOM 17, CKIT 1, CKIT 2, CMYZ. And apart from this, we have also used single stranded DNA and double stranded DNA for the studies. So with respect to the molecule QNA from fluorescent emission spectra, it was noted that the compound QNA shows a significant enhancement in the fluorescence in the presence of HRAS1 antiparallel G quadruplex. So here we can see we have taken a blank reading, that is only without any G-force. So we have taken a blank reading of the molecule. So it is almost very low. The fluorescence intensity was very low. So as we have added different mm, as we took readings for different G-force, so we can see the graphs for their respective G quadruplexes. So here we can see that the Q&A shows highest enhancement in the fluorescence in the presence of antiparallel HRAS1 G quadruplex, and it also showed the enhancement over with antiparallel 22 AG G quadruplex. And in case of QNMF, so here also the QNMF probe show significant enhancement in fluorescence in presence of antiparallel 22 AG G4 structure and antiparallel bomb 17 G4 structure. So hence, these two probes shows a sensitivity and selectivity towards antiparallel G quadruplexes over hybrid structures, parallel structures, single stranded DNA and double stranded DNA. So here this significant increase in the fluorescence along with the bathochromic shift or red shift that indicates that quinolinium and benzene parts of this these two molecules, that is QNA and QNMF compound, these are likely involved in the staking interactions with the G quarter plane of the DNA through pi-pi staking interactions. So the weak fluorescence observed in the molecular rotor. So here we can observe that blank, that is we have very weak fluorescence. So here a weak fluorescence, it is due to the dynamic motion around this vinyl bridge. So for that we have got a weak fluorescence. So one more powerful ways to study G quadruplex structures are that is by combining proton NMR spectra and F19 NMR analysis. And here the key distinction lies in how these techniques respond to the local environment. And hence, in proton NMR, the chemical shifts of the immunoproteins are highly dependent on their surrounding environment. So which becomes complicated in G-force because they adopt multiple conformations leading to overlapping and often ambiguous signals. So conversely, the F19 NMR is exceptionally sensitive to the electronic environment of a fluorine atom, and importantly, it remains unaffected by these local conformational variations of the molecule in the DNA backbone. So from this point of view, as one of our molecule, that is QNMF, has a fluorine 19 labeled probe. So this provides both high sensitivity and specificity for detecting subtle differences in G4 conformation, giving rise to distinct well-resolved peaks for each topology. So here we can see in our investigation, we have taken F19 spectra for in the presence of various G quadruplex sequences. That is, we have HRAS1, c-KIT1, BOM17, and even ssDNA. So these show that show different values for different what G quadruplex sequences. Hence, this dual functionality of QNMF, that is fluorescence enhancement and F19 NMR, so confirms that QNMF binds most effectively to antiparallel G quadruplexes and highlighting its potential as a powerful molecular probe or rotor capable of both detecting and discriminating between the different G4 topologies. So further, the limits of detection was calculated. So for both we have calculated. So for your illustration purpose, I have taken the QNA molecule. So here the binding properties of the QNA with HRAS1, antiparallel HRAS1 G quadruplex was carried out using fluorescence spectroscopy by varying concentration of HRAS. So the fluorescence spectra was recorded by sequentially increasing the concentration of HRAS1 while maintaining the constant proportion of QNA. So initially, the fluorescence spectrum with the zero equivalence of HRAS1 showed a very weak fluorescent signal at around 565 nanometers. So here we can see. So as the concentration of HRAS1 was increased, so with 0.05 equivalent increments. So the fluorescence peak at 595 nanometer intensified and exhibited was intensified and exhibited a red shift. So this red shift suggests that suggest a change in the energy level of the emitted light and indicates effective binding of the probe QNA with the G tetrad layer of the G quadruplex, that is antiparallel 22, sorry, HRAS1 G quadruplex. So further, the binding constant and uh, limit of detection was calculated for both the compounds, that is for QNA with HRAS1, the binding constant was found to be 0.99 into 10 raised to 6 per mole, and the binding constant for QNMF with respect to 22 EG, sorry, antiparallel 22 AgG quadruplex was found to be 0.49 into 10 raised to minus 6 per mole, and the limit of detection was found to be 1.01 nanomolar for QNA and 0.71 nanomolar for QNMF. So hence these can be detected in nanomolar concentrations. So further, with respect to QNMF, so the circular dichroism spectra was used to analyze the structure of 22 Ag. So here in the CD spectra, we can note that of 22 Ag. So here there is a small negative band at around 260 nanometers that indicates the formation of antiparallel G quadruplex structure, because this is the signature peak of the antiparallel 22 Ag G quadruplex. So further, additionally, there is uh, a split of positive band at around 400 nanometer that suggests the binding of the probe to the antiparallel G quadruplex. So here there are two curves. So one curve that is black in color, it is with only the reading was taken, or spectra was taken with only 22 Ag, and after that the probe QNMF was added to the 22 Ag G quadruplex and the reading was taken. So here the curve at that is 420 nanometer, 400 nanometer, which remains unchanged. So this suggests that the probe at 420, the peak at 425 nanometer, so which suggests that the probe is binding to the G quadruplex, antiparallel G quadruplex sequence. So in contrast, that is the hybrid, that is this 22 ECG quadruplex, when it is present in the KCl solution, it forms the hybrid. So here in the hybrid structure, that is in the presence of KCl, it exhibits a strong negative band at around 240 nanometer. So importantly, the positive band corresponding probe to the 400 nanometer remains unchanged, so strongly suggesting that the probe QNMF does not interact with this hybrid G quadruplex configuration. So further, the molecular docking studies were carried out using Autodock Vina software. So with respect to the molecule that is QNMF in the solution structure of human telomere 22 AgG quadruplex with the PDB ID 143 D. So here the docking interaction analysis revealed that there are predominantly pi anion interactions. So here you can see this is the G quadruplex structure, and this is the docked view of the compound that is QNA. So not QNMF, it is a QNA molecule. So this is a QNA. So it has interacted with the or G 22 AgG quadruplex. So here there are predominantly pi anion interactions were observed between the aromatic core ligand, that is aromatic sites of the ligand with the negatively charged guanine moieties. And collectively, these interactions suggest that the ligand adopts groove binding mode. So here the ligand is adopting the groove binding mode of interaction with the G quadruplex. So which is stabilized by predominantly pi anion contacts with the supplementary with supporting hydrophobic and hydrogen bonding contributions with the binding score of here the binding score was around 6.5 kilocalories per mole. So further, the life cell imaging or life cell bioimaging was carried out with respect to the compound QNA. So the life cell imaging was carried out using NIH3T3 fibroblasts using fluorescence microscope imaging. So here these are the results, that is the images. So the results revealed that the test compound, that is QNA, efficiently permeates cells and exhibits both cytoplasmic and nuclear localization. So here the merged images show partial overlap. So these are the merged images. So here the merged images shows the partial overlap of the red probe with the blue nuclear stain, so confirming that the molecule is capable of penetrating the nucleus. So these findings indicate that the compound is cell permeable and can also reach the nuclear DNA and may also engage with the cytoplasmic RNA G-force. So supporting its utility as a probe for intracellular G-quadruplex visualization. Conclusion. So the fluorescent probes QNA and QNMF possesses molecular rotor properties were synthesized, and the QNMF molecular rotor showed the highest quantum yield in the glycerol and significantly brighter fluorescence in a viscous environment. So the QNA and QNMF both showed high sensitivity and selectivity towards anti-parallel G quadruplexes in nanomolar range over parallel hybrid G-force and other single stranded and double stranded DNAs. And here the QNA, when it binds to the anti-parallel HRAS1, it exhibited red shift in fluorescence, that is from 565 to 595 so nanometer, that is up to 20-fold enhancement in the fluorescence. And with respect to QNMF, so here this QNMF binds to the 22 HG quadruplex sequences over other DNA sequences, and the detection limit was found to be 0.71 with 40-fold enhancement in the fluorescence intensity. Thank you.
Thank you, sir. Questions are the gateways to deeper understanding. The floor is now open for your queries. So, question from our side. Can I, sir?
Yes, ma'am.
Sir. Is stabilizing G4 always beneficial, or it sometimes disturb normal gene regulation?
It depends on the where this G4 structure is present, ma'am. Especially when these G4s are present in the oncogene promoter regions. So here stabilizing these G quadruplexes is very crucial, because these G4s interfere in the telomerase activity. So if the telomerase activity is disrupted or disturbed, so then the uncontrolled cell division can be controlled. So for this purpose, stabilizing these G-force is very beneficial.
Thank you, sir. Sir, one more question. The G
Yes,
quadruplexes mainly used as drug targets, or else they can also be used as drug carriers. Which one is more beneficial, sir?
actually, these are used for what for, as a. Can you repeat the question, ma'am, please?
Sir, how come this G4 quadruplexes mainly used? Either it is at a drug targeting the cancer cells, or else a drug carriers supporting
They
the
are
drug.
used. They are used in drug targets, ma'am.
Drug targets. Okay, sir. Sir, is any questions from the participant side? Sir, one more. You are showing in vitro studies. Is there any plan to do in vivo studies to support these findings?
In vivo studies we have, ma'am. Actually, the one molecule that is QNA what we have. So we are extending this work further by binding this QNA molecule with the actual nucleoside, that is G nucleoside, through one click chemistry, and further research is going on, ma'am.
Okay, sure, sir. Thank you so much, sir. So,
Yes,
as no queries from the participant side, we conclude the question and answer session. So, on behalf of all participants, I extend our heartfelt thanks for the way you patiently listen each question and provide a detailed answer. So, thank you, Dr. Mahesh Akki. The clarity with which you unrolled such a specialized topic has not only deepened our understanding of fluorescent probes, but also opened a new perspective in detecting the G quadruple structures. So with insights into that QA&M of preparation, drug applications you carried out so far, make us
Yes.
curiosity to explore how these frameworks may transfer the future medicine. Actually, it is a perfect blend of the advanced chemistry and the real world
Mahesh.
research relevance. Thank you so much, sir. Heartfelt thanks.
Thank
So
you.
now it's time for gratitude. So gratitude is the music of the soul, soft, humble, and deeply moving. So today's session stands complete only when we take a moment to honor and appreciate the minds and the efforts behind it. To do so gracefully, I invite Professor Sabri sir to deliver the vote of thanks.
Thank you, ma'am. I'm very grateful for giving me this opportunity. We extend our sincere thanks to our chief guest, Dr. Mahesh Shakti, for sharing their valuable insights and enriching our knowledge. We are grateful to the management and principal for conducting this wonderful faculty development program. And we sincerely thanks to the participants from various institutions and industries for their involvement and cooperation. And once again, thank you all.
Thank you so much, sir. Let's carry this energy forward as we meet again tomorrow for day 4 of our FTP. The same time, same platform with fresh perspectives waiting for us. Thank you all. Feedback link is posted in the chat box. Kindly fill it out and submit. Thank you, participants.
Mahesh Shakti, sir, thank you so much, sir, for your valuable lecture for us, sir. Thank you so much.
Thank you, sir. It's okay, sir. Actually, I have to thank you for providing me an opportunity to deliver my work in your FDP, sir. Thank you very much, sir.
Thank you, sir. Thank you.
It's okay, sir.